Hello everyone, it's Dr. Mohib, and in this lecture we will be going through gluconeogenesis, pyruvate kinase deficiency. As I have mentioned in last lecture, that we will be going through pyruvate kinase deficiency, but I have missed this topic, so we will be covering this topic in this lecture. Then we will be going through pentose phosphate pathway, glucose 6-phosphate dehydrogenase deficiency, electron transport chain, different types of shuttles used by NADH, and at last some memory drills. So first of all, gluconeogenesis. From at this point, you all must be familiar with gluconeogenesis and its different enzymes. So this is a sort of revision for you people. So if you look at the basic reaction, the glycolysis was the reaction where a molecule of glucose was converted into two molecules of pyruvate, and gluconeogenesis was the reverse of it, where the molecules of pyruvate are converted back into glucose. The process of glycolysis occurred in cytoplasm. Most of the reactions of glycolysis are reversible. If you look at the conversion of 1,3-bisphosphate into 1,3-biphosphoglycerate into 3-phosphoglycerate, they can go in either direction depending on the concentration of the two reactants. But there are some irreversible steps. For glycolysis was the hexokinase or glucokinase, or the conversion of glucose into glucose 6-phosphate, then the conversion of fructose 6-phosphate into fructose 1,6-bisphosphate. It was again irreversible, same was in reverse direction, and at last it was the conversion of phosphoenol pyruvate into pyruvate. It was an irreversible step. Now there was no such step where pyruvate can be converted into phosphoenol pyruvate. So for this, pyruvate must be converted into oxaloacetate by an enzyme pyruvate carboxylase using biotin as a cofactor. So a 3-carbon pyruvate is converted into 4-carbon oxaloacetate, and pyruvate enters the mitochondria. From there, the oxaloacetate can be converted into phosphoenol pyruvate by an enzyme phosphoenol pyruvate carboxykinase. From there, it can go up till fructose 6-bisphosphate. All of these steps are reversible. Now the conversion of fructose 1,6-bisphosphate to fructose 6-phosphate is regulated by insulin and glucagon. The regulation or the conversion is dependent on fructose 2,6-bisphosphate. Here we have fructose 1,6-bisphosphate and fructose 6-phosphate. So for gluconeogenesis, fructose 1,6-bisphosphate must be converted into fructose 6-phosphate. Now, the conversion of fructose 6-phosphate into fructose 1,6-bisphosphate and the reverse of it depend on the concentration of fructose 2,6-bisphosphate, and the concentration of fructose 2,6-bisphosphate was dependent on the glucagon and insulin levels. So when the body had increased insulin levels, this led to activation of fructose phosphofructokinase 2 and inhibition of fructose bisphosphatase 2. This led to increased concentration of fructose 2,6-bisphosphate, which activated phosphofructokinase 1, and thus there was a conversion of fructose 6-phosphate into fructose 1,6-bisphosphate and glycolysis occurred, while the reverse was dependent on the glucagon levels. So what did glucagon do? It activated the fructose bisphosphatase 2 while inhibited phosphofructokinase 2. This decreased the levels of fructose 2,6-bisphosphate, which in turn leads to the inhibition of phosphofructokinase 1 and activation of fructose bisphosphatase 1. This leads to the conversion of fructose 1,6-bisphosphate into fructose 6-phosphate, which can go on to take part in the gluconeogenesis. And the last step was conversion of glucose 6-phosphate into glucose, for which the enzyme used was glucose 6-phosphatase, and this enzyme is present in certain organs, like it is predominantly present in liver and also present in kidneys as well as intestinal cells or epithelium. So only these organs can take part in gluconeogenesis. In other organs, for instance in muscle, this glucose 6-phosphate can be diverted into glucose 1-phosphate, or in other words, this glucose 6-phosphate can be stored as a glycogen in muscles. So let's review the first aid. So gluconeogenesis. All of these steps are irreversible or the irreversible enzyme. All enzymes may be subject to activation by glucagon in fasting state. So if you look at the enzyme pyruvate carboxylase in mitochondria, the gluconeogenesis takes place in both cytoplasm as well as mitochondria. So pyruvate carboxylase converts pyruvate into oxaloacetate, and for it requires biotin and ATP, as well as it is activated by acetyl CoA. If we look at over here, the conversion of pyruvate into acetyl CoA took place by pyruvate dehydrogenase, and the increased levels of acetyl CoA, which showed that the cell has an increased ATP, led to the inhibition of pyruvate dehydrogenase and activation of pyruvate carboxylase. So the pyruvate carboxylase was activated by acetyl CoA. Then we have another enzyme, phosphoenol pyruvate carboxykinase. It was present in cytosol and it converted oxaloacetate into phosphoenol pyruvate, and it requires GTP. And GTP, where does it come from? It comes from the TCA cycle. When succinyl CoA is converted into succinate, we have GTP being produced in this reaction. Then we have fructose 1,6-bisphosphatase 1. It is also present in cytosol, which converts fructose 1,6-bisphosphate into fructose 6-phosphate. Citrate levels or increased citrate levels activates it, while AMP and fructose 2,6-bisphosphate inhibits it. Both of these shows that cell has decreased energy. So in decreased energy state, there is no need for glucose production, while the citrate or increased level of citrate shows that the cell has increased energy. Then at last we have glucose 6-phosphatase. This enzyme is present in endoplasmic reticulum, which converts the glucose 6-phosphate into glucose. It occurs primarily in liver, serves to maintain euglycemia during fasting. Enzymes also found in kidney and intestinal epithelium. The deficiency of the key gluconeogenic enzymes causes hypoglycemia. Muscle cannot participate in gluconeogenesis because it lacks glucose 6-phosphatase. Odd-chain fatty acids yields 1-propanol CoA during metabolism, which can enter the TCA cycle as succinyl CoA, undergo gluconeogenesis, and serves as a glucose source. While even-chain fatty acids cannot produce new glucose since they yield only acetyl CoA equivalents. Now, I have already mentioned in the TCA cycle lecture that first of all, we have to understand the beta oxidation of odd and even chain fatty acids. In beta oxidation, for instance, we have an even chain fatty acid containing 15 carbon units, while an odd chain containing 15 and even chain containing 14 carbons. In beta oxidation, this chain is cut while 2 carbon acetyl CoA are being released. So what happens is that for even chain, we will have, for 14 carbon, we will have 7 acetyl CoA being produced, while for odd chain we will have 6 acetyl CoA, a 2 carbon structure, and 1 propanol CoA, a 3 carbon structure. So this propanol CoA can enter the TCA cycle and take part in gluconeogenesis. So how does it? So as we can see, the different reactants or the products of TCA cycle, the oxaloacetate can be converted into phosphoenol pyruvate, which can enter the gluconeogenesis and become glucose. The acetyl CoA cannot be converted into glucose because it does not increase the net carbons of TCA cycle. The two carbons that enter as acetyl CoA are released in the first 2 steps of TCA cycle. But the propanol CoA, it is converted into succinyl CoA, which can increase the net carbon of TCA cycle and thus enter the gluconeogenesis. As evident by this, the odd chain fatty acids as well as some different amino acids remembered by the mnemonic WOMAT like valine, methionine, isoleucine, threonine, and pyrimidines all are converted into propanol CoA, which are converted into methylmalonyl CoA and into succinyl CoA, which can be converted into oxaloacetate, and oxaloacetate can be converted into glucose. So this was all about gluconeogenesis. Now let's go through the pyruvate kinase. Pyruvate kinase, the 13 number enzyme, it converts the phosphoenol pyruvate into pyruvate. So what happens when we have the pyruvate kinase deficiency? It is an autosomal recessive disorder, and what are affected? RBCs are the most affected. Why? Because RBCs do not have mitochondria and they use anaerobic glycolysis as a major energy pathway. So when the RBCs have pyruvate kinase deficiency, they will have decreased ATP, and they need ATP to maintain membrane integrity. The biconcave structure of RBC is maintained by the ATP. So when RBCs will not have pyruvate kinase, then as a result of pyruvate kinase deficiency, they will have decreased ATP, they will have membrane or structural changes, membrane failure. This will lead to the phagocytic removal by the splenic macrophages. So how does it present? It presents as in newborn as extravascular hemolysis as well as splenomegaly. And the disease severity depends on the enzyme activity. So this was all about pyruvate kinase deficiency. Now let's switch toward pentose phosphate pathway. Now pentose phosphate pathway, also known as HMP Shunt or 6-phosphogluconate pathway. It is divided into two reactions, and oxidative reactions and non-oxidative reactions. The oxidative part is the irreversible part, while the non-oxidative part is the reversible part. It yields two important structures or important molecules, an NADPH as well as an NADPH and ribose 5-phosphate. The reaction is as follows. The glucose 6-phosphate is converted into 6-phosphogluconate by an enzyme glucose 6-phosphate dehydrogenase, and as a result NADPH is produced. Then 6-phosphogluconate is converted into ribulose 5-phosphate and NADPH is formed and CO2 is released. The 6-phosphogluconate is converted into ribulose 5-phosphate, a 5-carbon structure. Then ribulose 5-phosphate is converted into ribose 5-phosphate, and which can be converted by an enzyme transketolase into fructose 6-phosphate and glyceraldehyde 3-phosphate. It is a complex process. You don't need to understand or remember all the processes. So the key processes or the key enzyme or reaction one have to remember is that for conversion of ribose 5-phosphate into fructose 6-phosphate, the enzyme is transketolase, which require biotin as a cofactor, and this enzyme is abnormal in, or the abnormal enzyme transketolase predisposes patients to Wernicke-Korsakoff syndrome. Korsakoff syndrome. Another thing to remember about these reactions is that both the pathways can work independent of each other. They can work independent of each other. So the pentose phosphate pathway provides a source of NADPH from abundantly available glucose 6-phosphate. NADPH is required for reductive reactions like glutathione reduction inside RBCs, fatty acid, and cholesterol biosynthesis. Additionally, this pathway yields ribose for nucleotide synthesis. Two distinct phases, oxidative and non-oxidative, both of which occur in cytoplasm. It is a purely cytoplasmic reaction, and no ATP is used or produced in this reaction. So the sites of pentose phosphate pathway are lactating mammary glands, liver, and renal cortex, which are sites of fatty acid synthesis or steroid synthesis, and RBCs. So the first product is NADPH of HMP Shunt. NADPH is used in various reactions. First of all, it is used as a reducing agent for fatty acids and cholesterol and steroid synthesis. So for this reason, it is present in lactating mammary glands. So for this reason, it is present in lactating mammary glands, liver, and adrenal cortex, because in all these organs, there is synthesis of fatty acid and cholesterol or steroids. Then another role of NADPH is it is cytochrome P450 system of liver. It is present in cytochrome P450 system of liver. Thus, it is also important in. Thus, it also occurs in liver. Then we have glutathione reduction and respiratory burst. And another product that we have of HMP shunt is ribose 5-phosphate, which is used for nucleotide synthesis. So this is nucleotide synthesis chart from first aid. The nucleotide consists of a 5-carbon sugar like ribose. Attached to it we have phosphate and base. So this is a structure of nucleotide. So base can be either purine or pyrimidine. So no matter what the base is, the ribose 5-phosphate is common for all of the nucleotides, and this ribose 5-phosphate comes from the HMP shunt. Then we have respiratory burst. The phagocytes like neutrophils and macrophages kill different type of bacteria and or bacteria by two processes. One is respiratory burst and another is using different enzymes. By respiratory burst, an oxygen is converted into superoxide radical using an enzyme NADPH oxidase, which converts NADPH into NADP, and thus oxygen is converted into superoxide free radical, and this superoxide free radical is converted into hydrogen peroxide using an enzyme superoxide dismutase, and the hydrogen peroxide is converted into hypochlorous acid by an enzyme myeloperoxidase, and this hypochlorous acid is used to kill bacteria. So all of this respiratory burst need NADPH, which comes from the HMP shunt. Now then we have glucose 6-phosphate dehydrogenase deficiency. If you look at over here, the very first step of HMP shunt, glucose need an enzyme glucose 6-phosphate dehydrogenase, which converts glucose 6-phosphate into 6-phosphogluconate. So when a person has a deficiency of glucose 6-phosphate dehydrogenase, he won't have the NADPH as well as the other yields of or other products of HMP shunt. But surprisingly, in patients who have glucose 6-phosphate dehydrogenase deficiency, the other reactions, the other HMP functions like nucleic acids and fatty acid synthesis are usually okay. So other HMP shunt activities are okay. Only one activity is compromised, the glutathione reduction activity of RBCs. So what happens in RBCs? In RBCs, we have a constant generation of hydrogen peroxide due to different stimulants, like when infection is going on or an inflammation within the body, we have generation of hydrogen peroxide within the RBCs, which leads to the denaturation and oxidation of globin chains of hemoglobin, which affects the RBC. Now, to neutralize this hydrogen peroxide into water, the cells need glutathione. What does glutathione does? That in its reduced form, the enzyme glutathione peroxidase, it converts hydrogen peroxide into water, but as a result, the glutathione is converted into oxidized form, the glutathione disulfide form. Now, to prevent or to carry on this cycle, we need the glutathione in its reduced form back. Now, for this, we have an enzyme in our body, glutathione reductase, or for conversion of glutathione from oxidized state into reduced state, the cell need NADPH. The glutathione reductase converts the oxidized glutathione into reduced form, and by doing so, it converts NADPH into NADP. And for, as we have learned earlier, that for constant generation of NADPH, we need glucose 6-phosphate dehydrogenase, which converts glucose 6-phosphate into 6-phosphogluconate, and in doing so, we have generation of NADPH. So NADPH is necessary to keep glutathione reduced, which in turn detoxifies free radicals and peroxide. Decreased NADPH in RBC leads to hemolytic anemia due to poor RBC defense against oxidizing agents. So what are the oxidizing agents for RBC, or the triggers for glucose 6-phosphate dehydrogenase deficiency? Now, oxidizing triggers. So the first thing is inflammation or infection. So whenever a patient deficient of glucose 6-phosphate dehydrogenase deficiency has inflammation or infection going on, he will have these symptoms, or he will have remission of the symptoms. Then we have another trigger as oxidizing agents like fava beans. And at last we have some drugs. And there are different drugs. So the drugs are, the first category are antibiotics. There are various drugs, with sulfur drugs being the most notorious to cause relapse of glucose 6-phosphate dehydrogenase deficiency symptoms. Sulfur drugs. We also have nitrofurantoin. We also have isoniazid, dapsone, and fluoroquinolones. These all are drugs or antibiotics which can precipitate hemolytic anemia in glucose 6-phosphate dehydrogenase deficient patients. Other drugs are antimalarials. Although this deficiency of enzyme is malarial protective, but there are certain instances, or the patient may have the deficiency of this enzyme but still may caught malaria. So the drugs for these are primaquine and quinidine. And at last, acetaminophen and aspirin can rarely cause hemolytic anemia in these patients. So oxidizing agents like fava beans, sulfonamides, nitrofurantoin, and primaquine, infection being the most common, can also precipitate hemolysis. Inflammatory response produces free radicals that diffuse into RBCs, causing oxidative damage. It is an X-linked recessive disorder, meaning that it affects only males. It is the most common human enzyme deficiency, more prevalent among descendants of populations in malaria endemic regions like sub-Saharan Africa and Southeast Asia. It is believed that this is an evolutionary process protective against malaria. We have Heinz bodies, which is denaturated globulin chains which precipitate within the RBCs due to oxidative stress, and bite cells are the result from the phagocytic removal of Heinz bodies by splenic macrophages. And for diagnosis of G6PD deficiency, we need a spot fluorescent test. Fluorescence spot test, which detect generation of NADPH from NADP. So the test is positive if blood spot fails to fluorescence under UV light, or it does not produce the fluorescence. Now let's review Heinz bodies and bite cells from first aid. Heinz bodies are present in G6PD deficiency. This is RBC and this is the Heinz body. It is actually the denaturated and precipitated hemoglobin. Since it contains iron, it requires supravital stain crystal violet to be visualized. So this cell is stained with supravital stain and it has turned blue due to the reason that it contains iron, and the phagocytic removal of Heinz bodies results in bite cells. The bite cells are also known as degmacytes due to removal of Heinz bodies by splenic macrophages. So when these oxidized or denatured hemoglobin precipitate or settle down in RBC, so what does the splenic macrophages does that it removes them or eats out this portion. So we have a structure like this RBC is formed. Now at last, let's go through the electron transport chain. electron transport chain and oxidative phosphorylation. Before going into electron transport chain, I have to go through the phosphorylation. Phosphorylation is the process of generation of ATP from ADP, and it occurs via two processes. So phosphorylation can be substrate level phosphorylation and oxidative phosphorylation. In substrate level phosphorylation, the phosphate group is transferred to ADP from a high energy compound, and in oxidative phosphorylation, the ATPs are generated using electron transport chain. Like for example, in conversion of 1,3-bis-biphosphoglycerate into 3-phosphoglycerate, we have generation of ATP. So this ATP being generated is substrate level phosphorylation, while the ATPs generated in electron transport chain are oxidative phosphorylation. Now, before going further, one have to understand that the electron transport chain consists of different complexes having different names. The complex 1 has another name, NADH dehydrogenase. Complex 2 succinate dehydrogenase. Complex 3 cytochrome BC1 complex. Then we have complex 4 known as cytochrome C oxidase. And at last complex 5 also known as ATP synthase. So what happens in electron transport chain is that electrons are transferred across these complexes, and in doing so, or during this process, the hydrogen ions are pumped into the intermembrane space. So this is the mitochondria consisting of outer membrane and inner membrane. This is the outer membrane, inner membrane, the mitochondrial matrix. So this is the zoomed picture of mitochondria. This is the mitochondrial matrix. Then we have inner mitochondrial membrane, intermembrane space, and over here we have outer membrane. So what happens in electron transport chain, which is present along the complexes are present along the inner mitochondrial membrane. So what happens is this: electrons are transferred from complex 1 into coenzyme Q. From here they are transferred into complex 3. From complex 3 they are transferred into cytochrome C. From cytochrome C they are transferred to complex 4, where they meet with oxygen and hydrogen ions to generate oxygen. And in doing so, when electrons move across these complexes, they pump electron from the mitochondrial membrane into intermembrane space, leading to the concentration difference or concentration gradient, with high concentration of hydrogen ions present in the intermembrane space and low concentration of hydrogen ions in mitochondrial matrix, creating a gradient or electrochemical gradient across the inner mitochondrial membrane. Using this electrochemical gradient, the complex 5, also known as ATP synthase, generates ATP from ADP and phosphate. Then we have another complex, complex 2, which gives electron to coenzyme Q, and the further processes are same, but the complex 2 has decreased energy levels while complex 1 has high energy levels. Another key thing to understand is the cytochromes. Cytochromes are a class of proteins that contain heme, or in other words, iron plus or iron or surrounded by porphyrin rings, present same in hemoglobin. But the difference in cytochrome and hemoglobin is that the oxidative state of cytochrome is changeable, or it changes continuously. When iron accepts electrons, it is converted into Fe plus 2 state, and when it loses electrons, it is converted into Fe plus 3 state. So it constantly undergoes oxidative state changes, while in hemoglobin we have a constant level of oxidative state like Fe plus 2. the hemoglobin works only when the iron is in oxidative state of Fe+2. The Fe+3 state of hemoglobin, also known as methemoglobinemia, the hemoglobin cannot work properly. So this was all about cytochrome and hemoglobin difference. Now let's review electron transport chain. The NADH electrons are transferred to complex 1, while the FADH electrons are transferred to complex 2 at a lower level than NADH. Oxygen acts as an electron acceptor to provide energy. In complex 4, the passage of electrons results in the formation of a proton gradient that, coupled to oxidative phosphorylation, drives ATP production, and ATP hydrolysis can be coupled to energetically unfavorable reactions. Now, there are also chances, or there are also proteins which can, as I mentioned earlier, that there is electrochemical gradient consisting of high concentration of hydrogen ions in intermembrane space while low concentration of hydrogen ions in mitochondrial matrix. And using this electrochemical gradient, the complex 5 synthesizes ATP. So there are certain proteins which pokes holes in this or converts or aids the movement of hydrogen ions from intermembrane space directly into mitochondrial matrix without going through complex 4, like over here. And these proteins are known as uncoupling proteins. So what happens? This electrochemical energy is converted into heat, and no ATP is being produced, as this gradient or this electrochemical energy goes without or does not go through the complex 5. So uncoupling proteins found in brown fat, which has more mitochondria than white fat, produced heat by increased intermitochondrial membrane permeability. It poke hole and decreases the proton gradient. ATP synthesis stops, but electron transport chain continues. So this can occur through uncoupling proteins which are present in brown fat, and brown fat is present in newborns to maintain or to fight the cold and hibernating animals. Aspirin overdose can also cause uncoupling of oxidative phosphorylation, resulting in hyperthermia. Another thing to remember is that cyanide and carbon monoxides are both inhibitors of complex 4. So poisoning by either carbon monoxide or cyanide inhibits complex 4, and thus it inhibits the electron transport chain, but they do it differently. The carbon monoxide binds the iron at Fe+2, while cyanides bind at Fe+3 state. One NADH produces 2.5 ATP and one FADH2 produces 1.5 ATP. NADH electrons from glycolysis enter mitochondria via the malate aspartate or glycerol 3-phosphate shuttle. So now to understand what are shuttles or why there are different types of shuttles present in our body. So the reason is, if we come over here, during glycolysis, glucose is converted into pyruvate, and in doing so, 2 ATPs are generated and 2 NADH. Then pyruvate is converted into acetyl CoA by an enzyme pyruvate dehydrogenase. In doing so, we have NADH being produced, and TCA cycle, the acetyl CoA enters the TCA cycle producing 3 NADH and 1 FADH and 1 GTP. But the pyruvate or these reactions take place in cytoplasm, while these reactions took part in mitochondria. And the NADH can only be used, or it can generate ATP via electron transport chain, which is present in mitochondria. So for these NADH to enter the mitochondria, we have different types of shuttles, as NADH as mitochondria is impermeable to NADH. So we have two types of shuttles for this. One shuttle is malate aspartate shuttle and another is glycerol 3-phosphate shuttle. The malate aspartate shuttle is present in liver, kidneys and heart, while glycerol 3-phosphate shuttle is present in skeletal muscles and brain. So what happens is that, as we have studied, that oxaloacetate can be converted into malate, and malate can be converted into oxaloacetate depending on the NADH concentration. Oxaloacetate can be converted into malate, and malate can be converted into oxaloacetate. The malate, when we have NAD converted into NADH and NAD. So when malate is converted into oxaloacetate, so this is sorry, this is a reversible reaction. So when malate is converted into oxaloacetate, NAD is converted into NADH, and when oxaloacetate is converted into malate, NADH is converted into NAD. So when oxaloacetate is converted into malate, the NADH is converted into NAD. So thus malate enters the mitochondrial membrane. Once inside the mitochondria, the malate is again converted into oxaloacetate, and NAD is converted into NADH. Thus the cytosolic, cytoplasmic NADH has been converted into mitochondrial NADH. Now, as the oxaloacetate has been also moved into the mitochondria, so we will have to regenerate it, and by doing so, or in doing so, glutamate is converted into aspartate and oxaloacetate is converted into alpha ketoglutarate. This is about the regeneration of oxaloacetate. The alpha ketoglutarate is also permeable and can enter the cytoplasm as well as aspartate, and in cytoplasm aspartate is again converted into glutamate, which leads to the regeneration of oxaloacetate by alpha ketoglutarate, and the glutamate can again enter the mitochondrial matrix. Thus we have no change within the molecules in cytoplasm as well as the mitochondria, but the NADH has been moved from cytoplasm into mitochondria, where it can enter the or take part in the electron transport chain. The another pathway or another shuttle is glycerol 3-phosphate shuttle. In cytoplasm, the dihydroxyacetone phosphate is converted into glycerol 3-phosphate. This is this reaction. If we refer to the glycolysis, this is dihydroxyacetone phosphate into dihydroxyacetone phosphate is converted into glycerol 3-phosphate by an enzyme glycerol 3-phosphate dehydrogenase, the cytoxole, and in doing so the NADH is converted into NAD. Now what happens, the mitochondrial, the same glycerol 3-phosphate is again converted into dihydroxyacetone phosphate by mitochondrial glycerol 3-phosphate dehydrogenase, and this enzyme is linked with succinate dehydrogenase, which converts this NADH into the FAD into FADH2, which can give its electrons to coenzyme Q. This is complex 2, which can give to coenzyme Q, and then into complex 3, which can go to complex 4. So this was all about the different shuttles. Now aerobic metabolism of one glucose molecule produces 32 net ATP via malate aspartate, which is present in heart and liver, and 30 net ATP via glycerol 3-phosphate shuttle, which is present in muscle, as the malate aspartate shuttle uses NADH while the glycerol 3-phosphate converts it into FADH. Anaerobic glycolysis produces only 2 net ATP per glucose molecule. Now let's review the ATPs being produced during the glycolysis. So during glycolysis, glucose is converted into pyruvate, and 2 ATPs and 2 NADH are produced. So during glycolysis, and these NADH can enter either through malate aspartate shuttle, where they can be converted into 2 NADH, and thus they produce 5 ATP. And if they enter via glycerol 3-phosphate shuttle, they are converted into FADH, thus they produce 2 multiply 1.5, thus 3 ATP. So the net ATPs via malate aspartate shuttle are, if we add these two, so they are either 7 ATP or 5 ATP through glycolysis. Then pyruvate is converted into acetyl CoA, and in doing so we have NADH being generated. Since we have two tons of or two molecules of pyruvate, then these two molecules are converted, two molecules of NADH are generated, so 2 multiplied by 2.5 is equal to 5. So we have 5 ATPs generated at this level, and then we have the TCA cycle. We have total of 3 NAD, so which is equal to 3 into 2.5 is equal to 7.5. Then we have 1 FAD H2, which is 1 into 1.5, which is equal to 1.5, and 1 GTP, which is again equal to 1 ATP, so 1. If we add all of these, we have 10. So this is the ATPs being generated in TCA cycle for 1 acetyl CoA. So since we have two molecules of acetyl CoA, so we will multiply it by 2. So this becomes 2 multiplied by 10, it is equal to 20. So we have 20 at this level. Or so we have 20 plus 5, 20 plus 5 plus this is equal to 25. Then if the malate aspartate shuttle is used, we add 7, so it is equal to 32. And if glycerol 3-phosphate shuttle is used, then we will have 25 plus 5, which is equal to 30 ATP. This was all about the ATP generation. And at last, some memory drills. Cyanide poisoning inhibits complex 1, complex 2, complex 4 or ATP synthase, and the correct option is complex 4. Glycerol 3-phosphate shuttle is primarily used in, is it liver, heart, kidney or skeletal muscle? And the correct option is skeletal muscle. Pentose phosphate pathway provides ribose 5-phosphate for, is it for glycolysis, fatty acid synthesis, nucleotide synthesis or TCA cycle? And the correct option is nucleotide synthesis. Which drug can precipitate hemolysis in G6PD deficiency? Is it penicillin, nitrofurantoin, metformin or aspirin low dose? And the correct option is nitrofurantoin. Thank you.